Well, thank you for the invitation to come speak with you today about my research. And I'm going to speak actually a little more broadly than just about the epileptic encephalopathies. I'm going to talk about some work in epilepsy in general. But my goal is to kind of give you an overview of the approaches that we're using in my laboratory for gene discovery, which tend to be genomic approaches. So my background is really as a pediatric geneticist and genomicist. So I have training, my postdoc training was here at the UW with Evan Eichler in microdeletion syndrome discovery. So I started there and moved that approach that we were using into the field of epilepsy, which at the time was relatively, I would say, understudied from a genomics approach. So I'll start with a little background about the disease itself, so you know where we're coming from, and then talk to you about the projects in the lab. So epilepsy, which is really not one disease, it's many diseases, but in general, epilepsy is really characterized by recurrent, unprovoked seizures. And this is just an EEG shown here of a patient having an absence seizure. It affects 1 in 26 individuals. So it's a fairly common disorder. There was a recent report by the Institute of Medicine within the past year talking about kind of how prevalent epilepsy is and kind of the need for additional research into both the etiology as well as the treatment of epilepsy, because a large proportion of patients are not very responsive to the drugs that are available today. But there are about 50 million people affected worldwide. So epilepsy can be characterized in many different ways. I'm going to talk today about seizure types. So the seizure types can be generalized, affecting the whole brain, or focal, starting at a specific part of the brain. And focal seizures can then generalize as well, but we're really talking about the onset of the seizure when we talk about the type. Epilepsy is characterized by the age of onset. So my primary interest is in those early childhood onset pediatric epilepsies, but there are epilepsy syndromes that don't manifest until adulthood. And then the etiology. So clearly today we're going to focus on genetic epilepsies, but there are also many other causes of seizures, including traumatic brain injury, stroke, tumors, for example, and those are clearly have a different etiology, and we're not going to be talking about those today. Importantly, I think there are over 50 different specific epilepsy syndromes described, and they're based on those features that I just talked about. They're based on age of onset, the seizure type, the evolution of the seizures, and many, many different classifications. The classifications, there's a whole team of neurologists who decide the classifications. But it's just really to give you a sense that it's not just one disease, it's probably many diseases. And our hope is that by understanding the genetics, we can even do a better job of classifying some of these syndromes. So I'm going to talk about 3 broad classes of epilepsy today. The first is generalized epilepsy, and these tend to be syndromes that are somewhat milder. They have onset in childhood or adolescence and are characterized by absence seizures or myoclonic seizures, sometimes grand mal seizures. And they often resolve in adulthood, although they will need treatment throughout the childhood years. They often get better over time. Focal epilepsies, there are many different focal epilepsies, and again, this just refers to where the seizure starts in the brain. I'm not going to spend a lot of time on that today because we're not focusing on it, but I'll give you an overview of what we know in relation to what we're doing in the lab. And then where we've probably spent the most time is on epileptic encephalopathies. Epileptic encephalopathies are severe early onset disorders, and they're associated with a poor developmental outcome. And we'll talk a little bit more in detail about those later. But some that you might have heard of include infantile spasms and Dravet Syndrome, which is one of the most common. So we've known for a long time that epilepsy has a genetic component. And this is known for hundreds of years, actually, since seizures were first recognized. And some of that data comes from twin studies. So if you look at twins, and this is actually a set of identical triplets, there's a high concordance rate, especially for the generalized epilepsies. And in fact, when you take identical twins or identical triplets for some of the generalized syndromes, you get very, very consistent age of onset for their seizures, response to drugs, et cetera. And then the other evidence comes from family studies. So there are families out there, large families sometimes, where there's a clear autosomal dominant inheritance pattern to the epilepsy syndrome in the family. And those have been used to do traditional linkage studies to map and discover the gene. In many cases for those families where the gene is discovered, they may be the only family which has mutation in that gene. So the genes discovered in this way have not yet had a very broad, have not contributed broadly to the genetic etiology of epilepsy, but it's always the hope that when you discover a gene in this way that you can expand to other affected individuals. And then lastly, we know for some of the severe epilepsies that de novo mutations are seen in children who have those epilepsy syndromes. And so that, again, is clear evidence that new genetic change in the affected child causes disease, and that's a clear genetic syndrome. This listed here are just some of the genes that have been discovered and responsible for the different epilepsies. One of the most well-studied is probably SCN1A. This is a sodium channel gene. It's the cause of Dravet Syndrome, but also causes some milder generalized epilepsies as well. And as you'll notice, many of these genes are ion channel genes, so sodium channels, potassium channels, acetylcholamide channels. But we're now beginning to discover that it's not just channels, mutations in channel genes that cause epilepsies, but there are now structural brain genes being discovered, and I'll get to some new discoveries that we've made towards the end of the talk as well. So despite the fact that I have a nice list of genes up here, all of these genes probably explain maybe 5% of epilepsy. So there's still a lot that we don't know. And one of the approaches, well, the general approach that we have taken is to ask, well, how can we use genomics, now that we have the tools to do that, to try and get at the genetic etiology of epilepsy? So we've taken two general approaches in my lab, and the first started with copy number studies, so looking for deletions and duplications in patients with epilepsy, in hopes that we'd discover both causative deletions and duplications, but also so that we could use those genomic regions to identify genes that we think might be important more broadly. And then the second approach is using the next generation sequencing technology, which is every geneticist probably now is using in one way or another. It's a very powerful technology that I'll talk about in the second half of my talk, and we're using that in a couple of different ways to try and discover new genes. So the first part of the talk, I'm going to talk about copy number variants in epilepsy. Copy number variants, for those of you who aren't in that field, are just that. They're differences in the number of copies of a given chromosome sequence between two individuals. And we've known for a long time that particular deletions or duplications cause things like mental retardation syndromes or intellectual disability syndromes. Copy number variants are important in autism spectrum disorder. And epilepsy is a comorbidity in both of those and in many other related disorders. And so it was very reasonable for us to think that let's take a look and see if we can find the same type of pattern in patients with epilepsy as we do in intellectual disability and other neurodevelopmental disorders. So I'm going to talk about two broad types of CNVs that we look for in the lab. The first is recurrent CNVs. And recurrent CNVs are just that. They happen over and over again in unrelated individuals. And they happen because of a specific pattern of sequence on the chromosome. And that's shown in this cartoon. So this is a stretch of sequence with genes A, B, and C. And on either side is a large block of DNA. And these two orange blocks of DNA are nearly identical in sequence. So this might be 200,000 base pairs that's 99% identical. And the reason this is important is that when the chromosomes pair at meiosis, there can be mistakes in the pairing that happens here. So these two blocks they're shifted. They shouldn't actually be pairing, but they do. And if recombination happens when they pair like this, you can end up with a gamete that has a duplication by this recombination event here, or a deletion of the intervening sequence. And so this is one of the gametes here with the duplication of genes A, B, and C, and this is the deletion gamete. So because this event happens at meiosis, these tend to be de novo or new changes in the child. And deletions like this cause syndromes that you might know of, such as Prader-Willi syndrome, Angelman syndrome, 22q11 deletion syndrome. And as a class, they're a fairly frequent cause of disease. The other type of deletion that's important is non-recurrent deletions. So these are deletions that happen, or duplications, where the breakpoints are random. So as you can see, these red bars here each red bar represents a deletion in a different patient. The breakpoints of each one are different, but they're overlapping. And these deletions and duplications are important from a research perspective because we can use series of patients with overlapping deletions to hone in on the most important genes or even gene for that disease. So in this example that I have, this is from a paper looking for deletions of the SCN1A gene in a series of patients with Dravet Syndrome. And as you can see, all 7 or 8 of these patients, while some of the deletions are large and some are small, the only place they overlap lies right over the SCN1A gene. So if you didn't know beforehand that SCN1A was the gene for Dravet, you could use this type of information to hone in on that gene and then go sequence it in other patients. So both of these types of deletions lead us to new genetic etiologies for disease, and we are looking at epilepsy specifically. So this is from a study that we published in 2010, and it was kind of the first large study of epilepsy in general to ask, do copy number variants play an important role in this disease? And so in this study, we looked at just over 500 patients with a variety of types of epilepsy, although the majority were this milder generalized type, to ask, do we find deletions and duplications in patients that we don't find in controls? And I probably wouldn't be standing here if the answer weren't yes. We absolutely did. So in close to 10% of our patients, we found a rare deletion or duplication that we think might be related to the epilepsy in that patient. And we found both the recurrent type of deletions and duplications and the non-recurrent type. Importantly, we found a significant enrichment of 3 specific deletions in patients with generalized epilepsy, and they're listed here. I'll talk about these top 3 just briefly, but each of these were found in about 1% of patients. And although 1% may not sound like a lot, if you think that all the genes that we know about cause about 5 to 10% of epilepsy, this is actually quite a significant risk factor for disease in this particular disease. So each of these were found in 1% of our patients. And then we found a variety of other recurrent deletions that we knew were important for intellectual disability and autism that we also found in our epilepsy patients. So we think that they're related to disease. So I'll just talk briefly about one of these, the 15q13 Micro Deletion. So as you can see, this genomic region is flanked by those identical orange blocks that I showed you. And the deletion takes out about 7 unique genes in the genome. And one of these is particularly intriguing because it's a channel gene. So it codes for the alpha-7 subunit of the nicotinic acetylcholine receptor. So this had already been proposed as an epilepsy candidate gene, but mutations have actually never been found in the gene. We first described this deletion actually in patients with intellectual disability, the majority of which had seizures. But it's much more frequent if you take an epilepsy population than it is if you take a population of patients with ID. And now there have been several studies that confirm that this is clearly an important risk factor for generalized epilepsy. It's specific for generalized epilepsy, so patients don't develop focal epilepsy. They don't really develop epileptic encephalopathies. It's really a pretty specific risk factor as far as epilepsy is concerned. But it's more complicated than that. So this is an example of some of the types of pedigrees that we see in families with this deletion. And the important thing here is that what this tells us is that this deletion is a significant risk factor for developing epilepsy, but it's not the whole story. So we find cases where the patient inherited the deletion from a parent who never had epilepsy and is seemingly normal. So there are probably other genetic factors that are playing a role. And that's clear in families like this, where you might have 2 affected individuals, one with the deletion and one without. So although there's a very, very high risk for epilepsy if you have the deletion, we have yet to discover what the other factors are that lead you to having seizures. And to even complicate things further, I already told you that we first found this deletion in patients with intellectual disability. And it's clear now that deletion is a risk factor for not only intellectual disability, but for schizophrenia. And this was shown in 2 very large studies. Autism, probably some behavioral abnormalities, including aggression and epilepsy. And this pattern of kind of variable expressivity and risk for various related diseases is true for the 2 other microdeletions that are important for epilepsy. So deletions at 16p13 and at 15q11, same story. They're present in 1% of patients with epilepsy, but they're found in patients with related diseases as well. So I told you that we also found non-recurrent Cnvs in a significant percentage of our patients. And again, these are the Cnvs that are interesting to us because they tend to point us towards interesting genes and gene families that we want to explore further. And there's just some examples up here of the contactin-associated protein 2, which is an autism candidate gene. But here we have a deletion of 3 exons in a patient. Contactin-associated protein 4, which is related, obviously, to the first gene. And then an example of 2 overlapping deletions in the OTS2 gene, which, again, is called OTS2 because it's an autism candidate gene. So these are all genes that we put on our list to follow up in larger cohorts and look for other types of variants. So we moved from that kind of general study to looking specifically at epileptic encephalopathies because we think that because these are very severe diseases, they're going to tend to have de novo changes, which we look for specifically. And as I already told you, these are epilepsy syndromes that have an early onset in early childhood. Sometimes the patients are actually normal in the first few months or even years of childhood, but once they develop seizures, they have developmental arrest and sometimes developmental regression. The seizures are often hard to treat, and there's a very poor developmental outcome with intellectual disability usually. So this is what our cohort looked like. We had 315 patients, and this is just a list of some of the specific subtypes of epileptic encephalopathy that we looked at. And when we looked for copy number variants in our patient population, again, we found about 8% of patients had a copy number change that was not seen in controls, and therefore probably important for the development of disease. These are some of the take-home points of that study. In general, we found 8% of cases. Interestingly, we didn't find any of those 3 deletions we found in the generalized epilepsies in this cohort. And this really just speaks to the fact that these are different diseases. So the epileptic encephalopathies probably have a different genetic architecture than the milder generalized epilepsies. We did find an enrichment of large deletions in our patients, which is not unexpected. And we found both deletions of known epilepsy genes as well as identified some novel candidates that I'll tell you about. So this is one example of those overlapping deletions that I told you we like to find. So these are 2 unrelated patients who both have the same type of epilepsy, myoclonic astatic epilepsy, and they each have a large deletion on chromosome 7. And the deletions here are represented by the stretches of red bars. And as you can see, they overlap partially, though not entirely, and the overlapping portion contains only 6 genes. So that makes it quite interesting. Many genes are expressed in the brain, so the problem is that when you have even only 6 genes, most of them are good candidates for epilepsy. So many of these are good candidates. There are also good candidates outside of the overlapping region that had been already identified in the literature, but one of our patients' deletions didn't include that, so we're more interested in looking at the other candidates. Another interesting fact is that this family, this proband, we found the deletion in, but when we went back to look at the family expecting the deletion to be de novo or new because it's so large, we actually found that it tracked in the family with epilepsy. So there were multiple affected individuals with different types of epilepsy, but everyone that we tested so far has the deletion. So again, this makes us think that this is probably an important deletion for the disease. There's a calcium channel gene that's brain expressed in this region that we've looked for mutations in, haven't found any yet. We're still looking. There's also a G protein coupled receptor. So there are lots of interesting genes to follow up here. So we didn't find any recurrent deletions that I told you about before, but we did find 2 patients with the same recurrent duplication on chromosome 16. And this is of interest because this duplication, again, first was implicated in autism spectrum disorder is found in patients with schizophrenia, with intellectual disability, and here it's found in 2 patients with similar epileptic encephalopathies. This family also has a family history of autism and autism-related disorders, so we suspect that it's the cause of their autism spectrum disorder as well. So we found lots of non-recurrent deletions, and this is just to show you the one at the top is a deletion on the X chromosome involving the CDKL5 gene. This gene is known to cause epileptic encephalopathies when mutated, so just tells us that we're on the right track, that we're actually finding things that are meaningful. And then this deletion at the bottom I'll come back to later, but it's a large deletion on chromosome 15 and involves multiple genes. And while we initially suspected that one of these was our primary candidate, as I'll show you later, this one at the end, CHD2, turns out to be probably the responsible gene. So this is not our study. This is just a large study from a group at Duke looking at focal epilepsies. So again, doing the same type of study, looking for copy number variants in focal epilepsies. And they find basically the same thing, that large copy number variants are important in this disease. So, and again, they didn't find any deletions of that chromosome 15 region, further confirming that that is specific for a subtype of epilepsy. So just to summarize what we know so far about copy number variants in epilepsy, I hope I've convinced you that we know that they're important for the etiology of disease, and they're important across a broad spectrum of epilepsy subtypes, so generalized, focal, and epileptic encephalopathies. So I'm going to switch gears a little bit and talk about the second approach we're using, actually 2 approaches, using next-generation sequencing. So many of you may know, but for those of you who don't, in the past probably 5 years, novel technology has come along that has changed the way we do sequencing in the laboratory. And it's changed so much, and it's so much better than it used to be, that the methods that we used to use, called Sanger sequencing, which was used to sequence the first human genome, that first human genome took about 10 years to sequence and cost several $1 billion to do. Today, you can contact a company and send them your blood and have your whole genome sequenced for about $10,000 in a matter of weeks. So this has really revolutionized the field of genetics and genomics and how we approach disease and gene discovery. So the way that it's, so we're applying this to our epilepsy cohort and trying to use it for gene discovery. And one of the ways, and probably the most common way that this has been used over the past few years, is called exome sequencing. And the exome refers to the part of the genome that codes for proteins. So it refers to the exon portions of the genes, represented here in black. And using a variety of techniques, you can pull out those exons or protein coding regions of the genome. That's important because those regions represent only about 1% of the genome. So it's much, much more efficient than sequencing a whole genome. You can take the sequence that you get from those exon regions, compare them to the human reference sequence and look for differences, look for damaging mutations. And the hope is that once you've applied all of your filters, which we'll talk a little bit about, that you narrow down to that single genetic change in your patient that's causing their disease. It's been a very successful approach for many diseases, and a lot of nice stories have come out of the University of Washington as well as many other places. But the place that we've applied it is Dravet Syndrome, which I've already mentioned to you. So this is a specific epileptic encephalopathy that has a specific seizure evolution, seizure type, a specific kind of general history, and is associated with developmental regression and poor outcome. And it's an interesting disorder because probably at least 75 to 80% of cases that are identified are caused by a mutation in the SCN1A gene. But the remaining 20 to 25%, you can't find a mutation in that gene. So the question is, this is a pretty homogeneous phenotype. So the question is, what is causing that other 20%? So we've taken the strategy to sequence a total of 13 cases of Dravet syndrome using exome sequencing. And in 10 of those cases, we sequenced not only the proband, but both parents. And the reason for doing that is to be able to compare the patient's genome to the parent's genome and look for that, hopefully one new change in the patient that is likely to be causing their disease. And then using comparing all of the patients to each other, we can also ask the question, in which gene are there mutations in multiple patients? So our hope was that given that this is a pretty homogeneous phenotype, that we could do this quickly, come down to 1 or 2 genes across our cohort and solve Dravet. But it's not that easy. So for those of you who are interested, this is the analysis and filtering strategy that we used. So basically, we used tools that are publicly available for mapping the sequence reads to the human genome, for calling the variants or the differences from the human genome. And then we filtered for changes that were not present in 6,500 control genomes that are available publicly, and for changes that we knew were going to affect the protein. So missense, nonsense, splice site, and frame shift changes. We had pretty good coverage of all of the bases that we were interested in, with a mean depth of coverage of about 120. That means that every base was sequenced over 100 times. So I'm not going to go into all the details. This is still a work in progress. But what I can tell you is that 3 of those 13 Dravet patients who had been sequenced for mutations in SCN1A had mutations in SCN1A that were missed previously. So on the one hand, this is a very expensive test for us to do to diagnose SCN1A positive Dravet syndrome. But on the other hand, it tells us that we're probably actually sequencing the right patients, because clearly these patients have Dravet, and hopefully the rest of them will find important mutations. So we did find 1 patient with a de novo mutation in a GABA receptor gene. These are known to be important for epilepsy. And when we sequenced additional patients with Dravet, we found at least a 2nd patient with a mutation in that gene, and probably a 3rd. So we think this is a somewhat important gene for Dravet syndrome. STXBP1 is a gene known to cause other epileptic encephalopathies, including Ohtahara syndrome. But we found a de novo mutation in one of our Dravet patients. And when we sequenced additional patients, we found at least 2 more. So again, we think that this is expanding the phenotypic spectrum of STXBP1 mutations. And then we've identified some what we think are very interesting candidate genes. So 1 patient had a de novo mutation in calcitonin 1. This is a protein that's active at the synapse and in shuttling neurotransmitters. We found a mutation in NCANE3, which is important. NCANE2 has been seen in patients with ID and epilepsy. And then NOL11, which not much is known about, but it's the only de novo mutation in this patient. But when we've sequenced 500 additional patients with epileptic encephalopathies, we haven't found a single other mutation. So we're kind of sitting on this, not knowing what it means. And then in 1 patient, we didn't have any good de novo candidates, but we did find an inherited mutation in a known epilepsy gene, a mutation that's already been shown to affect the function of the gene. So we think that it's at least partially responsible for the phenotype in this patient. And then we have 4 patients that we haven't solved yet. So we're still working on it. And we haven't solved it because we haven't found good candidate de novo dominant mutations in these patients. So we're in the process of reanalyzing all of the data with new tools that have come online since a year ago that we first did this. And we have identified some additional de novo candidates by using this approach. And then we're also looking at recessive models. So even though we think this is a de novo disease, it's certainly possible that some of these patients have autosomal recessive disease. So the rest of my talk, I'm going to talk about a targeted approach that we've used that has been really exciting for us. So targeted resequencing just refers to, rather than sequencing the whole exome, which is certainly gotten cheaper, but at over $1,000 a patient, we still can't sequence 500 patients' exomes, at least I can't, efficiently or cost effectively. So we have taken the approach to say, well, we have all this data. We have copy number changes in patients with epilepsy. They have important genes in there that we think are good candidates. And we have data from our Dravet syndrome exomes that we think are good candidates. So why don't we pull out those genes that we think are the top candidates and sequence them in a large cohort? So that's exactly what we've done. And the method that we've used to do this has been a collaboration with Jay Shinduri in Genome Sciences, who really developed and adapted this method for targeted capture. I'll refer to it as MIPS for molecular inversion probes. But this is just one of the methods to pull out the regions of the genome that you're interested in that's available. Basically, what you do is design primers that sit down and flank each exon or region of the genome that you're interested in. They bind to it. They fill in the gap there. And then you end up with all of your regions of interest. You do a multiplex PCR to amplify all of those regions. And at this step, you can add a barcode so that you can barcode each patient and then pull the patient samples and sequence them all together. And obviously, we use next-generation sequencing to do the sequencing part. So the project that we've been working on for about the past year is to look at 65 genes. 18 of these are genes that we know cause epileptic encephalopathies, and 47 of them are what we think are good candidate genes for causing epilepsy. We used about 3,000 of these MIP primers to pull out all of the exon regions of those genes. And then we sequenced 500 cases of patients with various epileptic encephalopathies to ask the question, can we find mutations in these known and candidate genes to diagnose these patients? And using the known genes, the goal there was that most of these patients hadn't been sequenced for every known epileptic encephalopathy gene. Some of them had been sequenced for SCN1A or STXBP1. But we wanted to ask the question, can we find mutations? And if we find mutations, what does it tell us about all of the phenotypes that are associated with mutations in those genes? So genotype-phenotype study. And then the candidate genes, obviously, we're looking for new genes that are important for epilepsy. So this is, basically an expansion of the cohort that we used for our copy number variant studies. But we had a total of, believe it or not, exactly 500 patients when it came down to it at the end of the day. And as you can see, there are some more common epileptic encephalopathy syndromes, including infantile spasms, Lennox-Gastaut, and early onset epileptic encephalopathies, and some that are more rare. But we wanted to look across a broad spectrum. So selecting the genes happened this way. We selected 18 known genes, and they're listed here in this box on the left. All of these genes are known to cause one or more epileptic encephalopathy syndromes. So they're fairly well-established genes. And then we selected 47 candidate genes, and those candidate genes really came from two approaches. This set of 14 genes here in the middle are genes that have already been shown to cause intellectual disability or maybe autism, and often with that, the patients would have seizures, but they'd never been looked at in an epilepsy cohort. And some of those genes, some of those genes, mutations in them cause milder forms of epilepsy, like generalized epilepsies. So we questioned whether some of those genes, mutations in those genes might cause more severe epilepsies. And then the second set, the larger set, was 33 genes, and those all were pulled from copy number variant regions that we had identified or that had been identified in the literature. And we used some bioinformatic tools to prioritize which genes in those regions were most important. We asked, were they related to known epilepsy genes? So we used a variety of approaches to select those genes because there were probably close to 1,000 to choose from when we collected all the data together. So our strategy here was to sequence the probands. So we sequenced all 500 affected individuals. We didn't sequence their parents. And then to look for rare non-synonymous mutations across all of the genes, and look for mutations specifically that weren't present in any of the 6,500 publicly available controls. And the reason for that is that these are pretty severe disorders. So we really weren't interested in changes that are present in normal individuals because we don't expect these changes to result in a normal phenotype. And then if we found a change in an affected patient, we then went back to look at their parents because we're really interested in finding de novo changes. So we had to know whether either parent carried the change. And to do this, we actually used the same strategy with targeted capture and sequencing, but we just picked out those MIPs that would pull out the region that has the mutation. So a MIP pick strategy. And some families, we had additional family members as well that we would test. So in general, we called a mutation pathogenic if it was a de novo change, so not present in either parent. If it was inherited, we generally called it a benign variant. There were exceptions to this where we had clear families with autosomal dominant SCN1A mutations, for example. So if the mutation segregated with disease, we would make a judgment call about whether it was pathogenic. So when you do this strategy, you want to make sure that you're actually sequencing the regions that you think you're sequencing. So this is just showing you gene by gene. So on top are our 33 candidate genes. On the bottom left are the known epilepsy genes, and the bottom right are the epilepsy-associated genes. And what this is showing you basically is the average coverage, or times that we sequence the exons in each of those genes. And overall, we actually required that a base pair be sequenced 25 times before we would make a call. And 91% of our target had 25x coverage across the gene. So pretty good results overall. So what did we find? Well, we started by looking at some controls to make sure that our strategy was going to work and that the MIP strategy was actually a good one. So we had 12 controls in our data set for which we had already exome sequenced. So we knew what variants they had. And there were 685 variants in these 12 individuals, and we had a 91% sensitivity. And the calls that we missed that should have been there actually just weren't captured by the MIPS. So that allowed us to know which MIPS weren't working properly. And then we had 24 additional patients where they'd had sequencing for one gene or another. We knew what the variants were. We picked up actually 100% of the variants in those patients. And then I'm not going to talk about this, but we're trying to develop strategies to actually find copy number changes in the genes by using the sequencing strategy. And we had 15 controls in our set that had deletions or duplications in one or more of the genes. We actually could detect all of these by looking at the depth of coverage or the number of times that gene was sequenced. So it works, but we're doing a lot more studies to figure out exactly how we can detect those that we don't know are there. We think we can do it, but we're not very good at it yet. So we had obviously we had known genes in our panel, and we found 34 patients who had a pathogenic mutation in one of those known genes. So these patients were previously undiagnosed, and we were able to find the mutation causing their disease. So this accounted for about 7% of our cohort. And I should say that all of the patients in our cohort, we excluded any patient with known mutations who had been evaluated by other methods. So although 7% is maybe a little lower than we wanted, this is a pretty selected cohort. And then we had 17 additional variants where we weren't able to look at both parents. So we don't know for sure whether they're de novo, so we can't decide yet whether they're pathogenic. Certainly some of them will be, so it's probably greater than 7% of our cohort that we've explained once we were able to get that information. So this is just a list of those 8 genes where we did find mutations in one or more patients. And as you can see, we found mutations in SCN1A. 4 of those patients are Dravet Syndrome patients, as expected, but we find some different phenotypes as well, which is interesting to follow up. And then I guess the other one of interest maybe is SCN8A. So SCN8A was first discovered by Exome Sequencing study, and there were some UW authors involved in that study as well. So there's only one patient so far reported in the literature, but we have at least one patient with a de novo mutation. And this patient's father has the same mutation, but because we're able to, because we sequence so deeply, we're able to identify the fact that only 13% of the father's alleles have the mutation. So he's actually mosaic. And we don't know yet whether he has any subtle phenotype, but we're looking into that. But we're pretty sure that this is pathogenic. And then we have some additional mutations where we haven't been able to look for the parents. So this is going to be a new, I think, important gene for epileptic encephalopathies. So more interestingly, we were really interested in our candidate genes because we thought we had some good ones. And we found 7 patients who had mutations in one of our candidate genes. And 2 of those genes had multiple variants in them and are, we think, new epileptic encephalopathy genes. So the first of these, and the one we're most excited about because we think it's truly novel, is CHD2. So we found 6 probands in our cohort, so about 1%, a little over 1%, with a de novo mutation in CHD2. And 4 of these were clearly damaging mutations that truncated the protein, and 2 of them were missense mutations. And if you remember, I showed you a large deletion in a patient from our CNV study where the breakpoint of that deletion was right in the CHD2 gene. And when we pulled information from the literature about 2 other patients with deletions in this region, the only overlap was involving the CHD2 gene. So that's the reason we selected it. And clearly it's an important gene within this region responsible for epilepsy. So CHD2 is a chromodomain helicase, member of the chromodomain helicase DNA binding family, of which there are at least 8 members. So it's thought to be involved in chromatin remodeling. And there are 2 other members of this gene family that are responsible for human disease. The first is CHD7, which causes charge syndrome. This is a multiple congenital anomaly syndrome that's associated with some developmental delays, occasionally epilepsy, but not always. And then very recently, so just in the past several months, studies from Evan Eichler's lab have identified CHD8 as an important gene for autism. So they found at least 6 or 8 de novo mutations in CHD8 in their autism cohort. So clearly members of this family are important for neurodevelopment, and we haven't looked yet in other neurodevelopmental syndromes for mutations in CHD2, but that's in the plans. So the patients in our cohort with mutations in CHD2 had a fairly similar phenotype. They all had seizure onset between 1 and 3 years of life. Median was 18 months. Most of them were normal developmentally prior to seizure onset, but not all of them. The primary seizure type was myoclonic seizures, but all of them had developed multiple seizure types across their lifetime. 3 of the 6 patients had photosensitivity, so photo-induced seizures, and 2 of them, I don't have the videos today, but our collaborator has videos of they self-induced seizures by standing in front of the television. So it's a clear, concrete phenotype. And we're interested in looking at, particularly at photosensitive patients to see if we can find additional mutations in this gene. And then they all had moderate to severe intellectual disability. So this just shows you a schematic of the gene and the spectrum of mutations that we found. So the 6 mutations we found are in red on the top. As you can see, they span the gene, and 4 of them are nonsense mutations. And the 2 underneath that I'm showing you are, one was reported in a patient with intellectual disability from an Exome study, and that patient had absence seizures in addition to intellectual disability. And then the second is a de novo mutation identified in an autism cohort. So again, already we're starting to see that mutations in this gene probably not only predispose to epilepsy, but just like the copy number variants, may predispose to a variety of neurodevelopmental diseases. And then the second gene in which we found multiple mutations is SYNGAP1. So SYNGAP1 has been previously identified in intellectual disability cohorts. There have been several mutations reported. All of the mutations in black on this figure have been previously reported in the literature. But all of them that are bold are patients with seizures. So we think that seizures is really one of the primary phenotypes associated with mutations in this gene. And the 5 mutations that we identified are in red. And as you can see, they're all nonsense mutations. So overall, we were able to diagnose 10% of our cohort with a mutation in either a known or a candidate epilepsy gene. And the blue bars here represent known genes. So you can see that SCN1A and CDKL5 are the kind of the 2 most common in our cohort. But SYNGAP1 and CHD2 fall right in there among the most common. So we think these are going to be important new genes for our phenotype. And then we have multiple genes, which I didn't talk about, where we found a single patient with a pathogenic mutation. So clearly these are rare causes of epileptic encephalopathies, but these are important to know about and still might tell us a little bit about disease mechanism. And then this is just showing you a breakdown by phenotype, in which phenotypes we found mutations. So I will summarize both parts of the talk. So first, the sequencing studies have been really, really powerful and are really changing gene discovery. And it's clear, not only from our studies, but from many, many that have been done now, that sequencing trios, so affected patient and they're both unaffected parents, is a really efficient way to identify de novo mutations and causative mutations. So we've applied this approach to Dravet syndrome. We think we have at least 2 genes that were not previously associated with Dravet but are important, and we're following up additional candidates. We've had a lot of fun with this targeted capture and sequencing approach. It's been, I think, really exciting to just pull out our favorite genes and be able to sequence them quickly and cheaply. So I didn't actually speak to that, but this is a very, very cost-effective approach that, depending on how many patients you do, if you do a lot of patients, you can sequence them for about $1 a gene. And we've identified what we think are 2 new genes for epileptic encephalopathy, accounting for over 2% of our cohort. And overall, we could identify 10% of patients with this approach. And then CNVs, I've really already summarized, but basically to remind you that CNVs are an important genetic cause of epilepsy and might be responsible for 5 to 10% of cases. So altogether, we think we're solving close to 20% of these cases. And importantly, both CNVs and genes that are important for this disease are important for other related phenotypes. And so they're telling us something about the shared genetic susceptibility for autism, intellectual disability, epilepsy, schizophrenia, and probably others as well. And all of these genes are candidates for follow-up. So our plan really is to expand this type of targeted sequencing study to look at some of those other phenotypes for our favorite mutations in our favorite genes. We're looking into doing functional studies of CHD2, for example, trying to figure out what are its targets. If it's a chromatin binding gene, what's it turning on and off? And then we're continuing with exome studies in select interesting epileptic encephalopathy phenotypes as well. So this is part of my group. There are a few more now. But I just especially want to thank Gemma Carville on the end here. She's a postdoc who's done the vast majority of this of the sequencing work. Which has been a collaboration with Jay Shinduri, who I need to acknowledge for sharing the technology with us early on. And then our collaborators in Australia, Ingrid Scheffer and Sam Berkovic, are responsible for collecting the epilepsy patients and providing excellent phenotype data. So that's been a really nice collaboration as well. And I'll take questions.
Thanks, that was a great talk. I'll start off with a question. I know that some of the types of seizures that folks can get tend to be associated with a certain type of treatment working better than another type of treatment. Right. Is there any correlation between any genes that are known now underlying epilepsy as being associated with perhaps one therapy working better than another, or is it thought that might come of this?
Right, so I'll just repeat the question. It was related to treatment, specific treatments, and do some treatments work better or worse for patients with mutations in specific genes, basically. So that is the goal, right, is to figure out what treatments work right away in patients with specific epilepsy syndromes. Probably the best example from what I talked about today is SCN1A, where you want to avoid certain sodium channel blockers because of the sodium channel mutation. So clearly the epilepsy gets worse if you use some medications with that. So it's one good reason for obtaining a genetic diagnosis in that cohort. Many of the others we don't really know yet, but that's the hope, is that you need to find the genetic diagnosis first before you can ask the question, does this certain patient population respond to this drug or not? So that's kind of what we're hoping to do. There are potassium channel mutations, so you can think about those type of channel blockers, calcium channel mutations, and many others. Yep.
I'm just wondering if you considered extending your targeted resequencing cap to include unexpressed mutations of genes. It sounds like you're very focused on exome. Is there a reason to think that splice variants or other kinds of mutations that can occur in introns don't play as much a role as exonic mutations, or is it just that exomes are easier to
right.
work with at this point?
So the easy answer is that exomes are easy to work with because we know how to interpret mutations in coding regions of the gene. So the question was, why aren't we looking at the non-coding portions of the gene? So we do look at the splice sites. That is included in our capture. But you're right, we don't look at introns. We don't look at regulatory regions. There's no reason not to, except that you have to think about how you're going to interpret those variants. So that's certainly something that we'll do eventually. And one of the things we would love to do actually in those Dravet patients that are SCN1A negative is to start looking at some of those non-coding portions, because it's, we're not finding a lot of other genes that are important. So it may very well be that they all have SCN1A mutations. We just haven't detected them yet.
very nice talk. Thank you so much for coming to speak to us.
Sure.
Could you tell us a little bit about epilepsy and mutations and when in development the pathogenesis begins that you get nervous system defects that then lead to either epilepsy or learning disability, and whether or not these studies are going to point us in the direction of genetic therapy?
I'm probably not the right person to ask that question. If you're asking about when, so the question was when in development do the changes happen that lead to epilepsy or other related disorders. So the genetic changes in all these cases are germline. So they're present from the get-go. So why some patients develop seizures on the first day of life and why some patients develop seizures at age 10 and some at age 38, I don't think is clear. Certainly there are some known changes in expression of some gene families where one gene is turned off at a certain point in development and another one takes over. GRIN2A and GRIN2B are like that, and they've been, some mutations have been identified in autism and epilepsy. But I don't think we know enough yet about all of that to be able to answer that question. Sorry. Yeah.
Are these abnormal channels expressed only in the brain? And is it possible that these channelopathies also have manifestations elsewhere? And some patients have seizures and others have other manifestations.
Right, good question. So the question was, are these channels expressed only in the brain or are they expressed elsewhere? And might you see other things? So they are expressed elsewhere, some of them. There's a low-level expression of SCN1A in the heart, actually. And there's a group looking at sudden unexplained death in epilepsy, which is a huge problem in these patients. They will unexpectedly die early. And one of the hypotheses is that there may be changes in cardiac rhythm that affect this. So there are some examples like that. They're not just expressed in the brain. Dravet patients also develop an abnormal gait, kind of in adolescence usually, kind of a hunched over crouch gait. It's not clear to me whether that's just because of central nervous system control or if there's some muscle expression that might be affecting that as well, because they get, they have some weakness as well. So there certainly could be other manifestations.
Thanks very much.
Yeah, you're welcome.